Okay, so in the last class we started. I gave you some historical things or the events that has happened in molecular biology. So today I will go in little bit more details and I'll show you how you can look at a couple of things, and then uh, uh, yeah, I'll take a specific example of HIV, and you'll see how things are happening there. Okay, uh, as a genome. Okay, so as I mentioned, the last 20 years or so, people are reading a lot of the genome of different species and collecting them. And so what they have learned actually, by the way, I mean, this is also a very interesting book. What is life actually? And then what are the life forms, the different kind of life forms? How many species are there on the planet? Okay. So this is a book by Schrödinger, where he talked about physical aspects of living cell. It's a very interesting book actually, although a bit old. In 1948 it was written, but it will give you a good insight. Schrödinger was a famous person, I hope you know. So how many life forms are there? Scientists have estimated there are about 1 trillion life forms on this planet. Right. This is a nice paper. So total microbial diversity, they have globally, they have predicted. Okay. So this gives a nice thing. And how many plants? So although this picture is a little bit old because they keep on changing some data, but it gives a good idea. There are about 31,128 useful plants, right? And about 369,000 flowering plants, right? And then there are vestibular plants for which complete genome is there, right? So all this classification is there in biology, right? So you can look at the plants and other things, right? Uh, so this is something nice. How many plants are there? So all this data you can access at the NCBI website. Then in useful plants, you see that for human food, something around 5,538. Medicine, 17,810. Fuels, poisonous plant, animal food, 3,649. Social use, right? So this is something, Indian medicinal plants, something they have listed around 5,000. So basically, so you observe the diversity in the nature and try to collect this information and then classify them, and also read their genome and put it on the website, right? So anybody can access this data. How many bacteria on this planet? This is something a very remarkable thing. Uh, so if weight of one blue whale is around 140,000 kilogram, all bacteria on the planet, the weight is equivalent to around 50 million blue whales. This is the amount of bacteria we have on this planet. They are very, very useful. And normally all these microbes, especially bacteria and viruses, they are designed for one ecosystem or they evolved in one ecosystem. So they become dangerous also, right? Because on your body there are a lot of bacteria, and some of the bacteria are helping you in digesting the food and other things. Many activities they do in your body, right? But the thing is that so any set of bacteria is useful for one species, right? So they live in their ecosystem, and they become dangerous when we change the ecosystem from one species to other species. Then they become disease for the other person, right? Because bacteria wants to survive there, and there is a fighting with the immune system and so on and so forth. All kind of things will happen there. Okay, so these are my favorites. Bacteria is very favorite, and I think, as you know, there is new branch of bacterial computing where you can use bacteria for computing different things. There is a bacterial camera also. You can take pictures. Okay, all kind of things are there with bacteria. Okay, there is a one society which which has competition every year to make art, different kind of art form using bacteria. So all kind of things are happening with bacteria. And people have tried storing data also on bacteria. We have done an experiment where we have stored some data on plasmid DNA of the bacteria, and we retrieved it successfully here in India at NCBS Bangalore. We have done this experiment. As you mentioned, see, there are a lot of bacterial infections that happens every time, All these diseases are happen with different part of your body, uh, you know, they may cause problem. So microscope discovered bacteria something around 1654 by this guy, right? And there is a actual note and how he actually created this microscope to look at these things. I think some of you might have seen the bacteria in, you know, in the yogurt or whatever curd. By the way, all this industrial evolution, I mean, that industrial revolution that happened in Europe, which is known as Age of Discovery. I think some of you might be knowing that, right? 1600 and so on, a lot of ships started coming from Europe to different part of world and kind of explore. They started exploring different parts of the world, and of course there was a lot of interaction with India. So earlier the interaction used to happen through land. So most of the stuff like spices and other things they used to go from Surat and Kerala and Calcutta to different part of world, right? So I mean they used to go to Arabic people, the Saudi Arabia and other people, the Middle East, and from Middle East to Europe, and from Europe to the other parts of the world, right? So in fact, all this happened because there is a ocean current in the ocean, which is because of this bacteria only. This bacteria actually controls the temperature of the ocean. So, and because of that, these variations, there are, cold current and hot current, right? So, which because of this, and I think the discovery of that resulted in that people who are able to, you see these pathways are available through which actually these ocean currents are there, and using, so if a ship is on this one of the ocean current, it requires less fuel, and then you can actually go for a longer distance, right? So these kind of things were discovered, and it is because of that a lot of things happened, and this resulted. So basically, it is because of that bacteria, this age of discovery, what is known as from 15th century to 17th century, people started going from ship, and one of those things, the bigger ship. I don't know whether you know Charles Darwin. Actually he traveled in this bigger ship, and so people say that it is at that point he, observed a lot of different diversity in the nature, right? And he collected a different kind of uh, plants, samples of seeds and other things from different locations in the world, and then he came up with this idea of evolution, that the theory of evolution, for which Charles Darwin is famous, that he wrote that book, Origin of Species, right? And I think people say that no, nothing makes sense uh, in the nature unless you look at from the eye of evolution. Evolution is a very, very important thing, right? So you might have read about evolution sometime back in your high school or whatever, right? So this is all, see, because of that bacteria, the ocean current, all the ships traveled throughout the different parts of the location, and that resulted into this book, Origin of Species in 1859. I think some of you should read this. Nice reading. For a Sunday afternoon, you can look at this original thing. Okay. And Darwin became quite famous actually after this. And then during this journey, there was this guy, Gregor Mendel. So when these ships used to come to Europe, Mendel job was to collect these seeds and other things at one place, and they were trying to do different experiments with those things, trying to create new kind of plants and so on, right? New species or something. There he learned about this. He did this famous pea experiment, right? And by taking different combinations and so on. Actually, the first time somebody observed that there is what is known as the idea of genetics or heredity. There are certain traits which transform from one generation to the other generation. Okay, so this was very nice thing which he observed. Although Fisher, who was a mathematician and a statistics person, Ronald Fisher, he later on gave the entire mathematical theory for this genetic things. Okay, so this was the beginning of genetics. It's all because of that bacteria, right? Ocean. Okay, so now life 1.0 now, or we all these genomes you can call it is software for life, right? So if you have one genome of bacteria, another genome of bacteria, so they are nothing but software or like a program, right? And you can modify these programs to make different things, right? So this is an old picture up till 2014. You can see that, you know, from 1995 to 2004, there was not much. By 2014, around these many genomes were there, but now it is much more. In fact, some of these things becomes obsolete also. They update different versions of the species and so on, so forth, okay, in the NCBI website. Okay, and they try to find relationship in terms of evolution, right? So for example, so people have observed that all the humans, okay, if you try to correlate their genomes, right, try to compare their genomes, see that out of 1,000 bits of ACGT, only one bit is different. Okay, so 99.9% all humans are same. Okay, so this is one observation. Another thing is that 70% of human genome is similar to sea urchin. Sea urchin is a species found in ocean, right? So all different kind of observation people are able to make by looking at this genome, right? This genomic data, and you can compare. So there is a tree that has been made by scientists, and there are many softwares available. For example, there is one software called Mega. So you just plug in these genomes, and it will do the analysis and will, you know, give you the chart. So some people are running companies also. For example, you know, if you want to find your ancestors, you can do that by just comparing the genome, right? From where is the origin? Okay, and I think with this genomic study, they found that the oldest human DNA that they were able to locate in terms of while comparing, right? Because it was in Chennai somewhere, near to Chennai in Tamil Nadu, basically, from all over the world. So I think these kind of things you can find out through genetics. A lot of things are possible because now you have the data. Since you have the data, you can actually play with it, right? And all kind of things you can do. Okay. Okay, so this is a bit much larger picture. You can compare. So basically what happens, for example, I think you must have looked at that. By the way, there is you know, there is a coincidence or whatever, a connection. In the Indian literature also, you might have seen that Bhagwan Vishnu has 10, this thing, right? Have you heard of those things? 10 avatars, right? Starting from Kurma avatar to, you know, different things, then Matsya avatar and like that. People try to relate with the evolution, actually. That is how it happened. So actually what happens, initially life is started in the ocean, right? That everybody knows, right? And then from ocean, it comes to, you know, earth. So there are species which were common, which can survive in ocean as well on the earth. This is how evolution actually took place. So what happens when a species is evolving? Based on the environment, remember, as I mentioned yesterday, environment makes certain changes inside your genome. Sometimes these changes are, you know, good, sometimes they are bad, depending upon the environment. If they are bad, it will lead to a disease. And they are good, you will actually survive in a different environment as well, right? Somebody who's, that's why actually in some countries, they ban transferring certain plants and so on. For example, in New Zealand, you cannot take a plant from anywhere and go to the New Zealand with that plant. You must be knowing that, for example, in the New Zealand, there is no snake. Snake-free country. Okay. Why there is a snake-free country? So there are different biodiversity in different parts of the world. But similarly, there is a island, I don't know where you know, there is a snake island, where per every square feet, you have around, I think, 6 to 7 snake. Okay. Full of snakes. So at one portion, there is no snake, other portion, there is, full of snake. This kinds of biodiversity happens because of the environment, right? Where they are. Okay. And those things change the different path for the species, right? So what happens, so people say that, for example, that humans evolved from monkeys, right? So at some point, genome modified. Some people remain monkey and the other will evolve into humans, right? Those kind of things take place, right? Depending upon the environment. Okay. This is a theory. These days, I think if you know, there is a recent, in the human genome also, X chromosome used to be very mysterious thing. And the first time people are able to, there is a X chromosome, Y chromosome, right? In each human, right? So this X chromosome, they are able to actually look at in more detail. And there are a lot of wonderful things there. Actually, if you really want to understand the human evolution, X chromosome you need to study, and it's huge. It's very large actually. Okay, so now let me show you. By the way, if you quickly want to know more about the genomes and how, for example, in the case of, you must have heard about SARS, right? Just like Corona, right? There was a SARS also. So people were able to trace. How do they trace origin of a disease? From where? So they collect samples of virus around. So a virus might be infecting this guy, another guy, another guy. So they try to collect the samples and then they compare the evolution of the virus. So for example, if you get a viral infection, which I think most of you have got in your life till now, each of these viral infection, so the virus that enters inside your body, after fighting with your immune system, evolves. Again, theory of evolution, because in that system it is evolving, right? And will adopt to the new environment. And then, so something that entered in, so the genome of that virus, which was on day 1, after 10 days, its infection will be different. In fact, and so how the viral actually do the replication, they take their genome and insert in your genome, they attach their own genome, and then they make copy of their own, right? So they will use your own machinery to make different copies of the genome, right? And in fact, because of this, now people know in the human genome, 2% genome is because of these viral infections. So their genome is inside our genome. So our genome is basically a playing ground. People have done a lot of experiments and interactions, and those things are there recorded in the genome, right? So all kind of things are there. So by the way, in this book, it's a very, very nice book, very small book, first of all. Second, there are a bunch of case studies. And using these case studies, you can observe. So for example, they were able to identify the origin of SARS, how it originated, because they sampled all these viruses, viral infections from different humans, and they're able to trace the story, how a person got it in Hong Kong, and then they shared a lift. In the lift, how many people were there? They compared that viral genome and so on and so forth. So the whole story can be created just by comparing the viral genome. Okay. So that's a very powerful tool. Just you are using your programs to look at the data and trying to find different connections. Okay. This is something really interesting. So just like each book has a accession number, most of these genomes have been assigned accession number. So I'll show you one of the first one. How do we look at it? If you go to here, I'll just go to NCBI. So this is the place where all genomes are there. And let me actually copy the first one. Say, for example, this is the accession number. This is the accession number for HIV. Okay. You go here, go to nucleotides, go here, paste it, and search. Right? So this is the complete genome. Normally these genomes are in two file formats, FASTA format and gene bank format. This is the gene bank format. So see, you can read this is 9181 base pair. The total length is 9181 base pair. ssRNA means single strand RNA molecule. This is RNA. So viral genomes are either made of RNA or DNA. Mainly the most of the infectious viruses are made of RNA. So genome is made of RNA molecule. So you know the DNA and RNA, what is the difference? In DNA we have ACGT, in RNA we have ACGU, right? So that is the difference. So you can see that when this sample was collected, this and that, all the details are there. And also, you see, this is the gene bank file. And this, you know, there are about 9 genes. What are genes? A portion of DNA which makes protein. So there are total 9 genes in HIV, which gives something around 45 to 50 proteins. Okay, so small. So you can actually try to look at it. Humans is very big, right? But here it's very small, so you can look at it. And these are kind of things. So geolocation is there. And look at this thing. These are alphabets which corresponds to amino acids. So this is a protein. A protein is combination of amino acids. So these are nothing but amino acids. So basically, you know, for example, this is the gene GAG-POL. There are all these genes have been named and so on and so forth, right? So these are the 9 genes are there, and so what happens? Some portion here, combination will give you another protein. That kind of things are there. So 45 to 50 proteins are known for HIV virus. Okay, so you can see, but total data is here at the bottom. You can see this is the data starting from here. So this is the gene bank format where you have all the data. By the way, you can see that TTT is there, all those kind of things are there, which we are trying to avoid in our DNA storage, but here those kind of things are there quite often, right? So this is the HIV genome. You can look at it starting from, so this is the format, right? 1, 2, 3, 4, 5, 6, right? These 6 columns, each is having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 bits at one column. And like that, you have 9181. By the way, different HIV viral genome have minor variation in the size also. Somebody may have something else, right? Because you know, it will evolve and change. So something around in the range of 9,000, between 9,000 and 10,000 base pair, right? That kind of thing will happen. This is HIV, right? So there is another format which is known as FASTA format. So this, you know, you have this gene bank file has a lot of additional information, when it was collected, what kind of proteins, this and that. But when you want to play with the computers, you just require this data, which is at the bottom. So there is a FASTA format especially created for that, which people are also using for DNA storage, those kind of FASTA format, right? So okay. And also, so this has a lot of browsers also. For example, you can look at so gene bank and so on. You can submit something here. And if I want to show you different species, you can look at it. And they have been arranged nicely. So if I say that genome, so I click the genome. So human genome, microbes, all these bacteria and viruses are there, right? All other kind of things. So human genome, if you click, you can find all these chromosomes. You see, these are 22 chromosomes, X and Y chromosome, and this is mitochondrial DNA, right? This is the whole collection. Yesterday, I mentioned to you chromosome number 7, and the top one is known as P region, the bottom one is known as Q region. P and Q region is there, right? Nomenclature is there. And just deletion of 3 bits of letter from chromosome 7, you get cystic fibrosis disease, right? So that kind of disease classification is there in terms of what is possible. So obviously I can click further and look at any data inside the chromosome. All these nice things have been. So people have spent so many times, so many companies like IBM, Google. In fact, Google wanted to do one thing at one point of time. They wanted to collect all human genome of all the population and then they have the whole control, right? They know everything about you in that case, right? Who you are, what you are going to do, because all kind of histories are recorded in the genome, right? People are even developing, although there are certain things like who is going to become criminal also. Can you predict that from the genome? Uh, all kind of things are there inside the genome, right? How you evolved, or whatever you took from your different ancestors, right? It has given you unique signature. Okay, so all kind of things are there. Basically, a lot of debatable topic, but you know, uh, a lot of things are possible. Actually, I would say that this is the ocean of data in front of you. You are lucky that you can see this data, and now you can write computer programs to do various things on this data, right? A lot of things are possible. Okay, so similarly, you can look at some of these other access. These are some of the great time when the genome was sequenced and they come the front cover of Nature and so on, from 1990s to 2003 for the whole human genome project when it was done. So actually, and I'll give you a brief explanation of how central dogma works from DNA to RNA to protein. This is a slide actually I have taken from the US government website from DNA to humans. You see, in the inside each cell there is a genome, those chromosome. If you chromosome unwind, you get this double helical DNA, and then there are genes which are instructions for making protein inside the DNA, right? So they will separate it, and then you can make protein by taking different combinations, different places, right? So if a body needs a certain protein, it will go to some portion of DNA from somewhere, another portion from another where, splice them, combine them together, and then they make protein. So that DNA string is converted into messenger RNA, and messenger RNA is converted into DNA using a map known as genetic code. That is the whole process, okay? And then some of these proteins combine together to form a supramolecular families of proteins. So two bigger tasks inside your body, okay? So this is how I should give you a mathematical description. So initially, this is very simplistic version of central dogma. So DNA, there is a process known as transcription. So DNA will convert it into messenger RNA. DNA is basically a string of ACGT. Okay. And the genes are, so basically in that DNA, some portion is taken from one place, another portion is taken from other place, another portion is taken from other place, splicing happens, and then the whole DNA is converted into messenger RNA. So where T is replaced with U, that's all. Right? And then there is a genetic code which works in translation to give you protein. So here it is, system here. So DNA is a string of ACGT, mRNA is a string of ACGU, and protein nothing but 20 amino acids. And genetic code is mapping from RNA space to protein space. So it's something like we have a digital signal, it is converted into analog signal. Because protein interact through structures. Okay, one protein interact with another protein, which is like a analog. And all that data is digital. Okay, so it's a digital to analog converter with genetic map is sitting here. I'll show you the genetic map. This is the map from messenger RNA. You take 3 bits. So suppose the bits are CUU. So all these red ones, LEU is leucine. Okay, valine. So all these are amino acids. Each amino acid can be mapped with different combination of ACGU. Okay, so for example, leucine, you see, CUU also give you leucine, CUC also gives you leucine, UUA gives you this, same thing, UUG will give you this, UUU will give you same thing, UUC will give you, no, sorry, that is not. 1, 2, 3. Yeah, so these are 3 ones, right? So leucine can be encoded with any of these 3-bit from mRNA. Okay, from the mRNA string, you read 3 bits, and if any of these 6 will come, you replace it with leucine. Leucine is the amino acid. You read next 3 bits and assign from this table the corresponding amino acid. So that way, one amino acid, another amino acid, another amino acid, you form the chain of amino acid, which folds into the 3D structure of protein. That is how the whole system works. A lot of paper has been written on this. This is a beautiful mathematical structure, genetic code actually. So there is a start codon, a stop codon. When there is a protein, actually, there is a start codon, a stop codon, right? In between, there is an instruction for making protein. There are companies which are actually optimizing even this also, they are doing all kind of different things. Okay. Anyway, I'll leave this. Okay, so I think we can do quiz, right?